I can say a little bit towards standards and provenance that in HubMap, we are using a W3C standard already, called the W3C provenance standard. So there's that piece, and then there would be linking up, and we've done some work already with the HCA to make sure that we're getting common elements in provenance.
So what kind of standard are they using for their data provenance?
I don't know that they are using this. They wrote their own, I think. Okay. Last I talked to them, I don't know, but they seemed interested in the W3C standard that we talked about.
Okay.
It's of course going to be their own implementation.
Mm-hmm.
But so we compared. I forget the name of the person I talked to now, but I could go look it up. But we compared like the main elements, data elements that we're collecting for provenance across both, and it seemed, you know, they had one that we didn't have, and we had one or two that they didn't have. And I said, okay, we're going to go add yours so that we can be, you know, and we did.
Yeah.
Add the, you know. So.
Okay, so maybe we could quickly go through some of these questions, because I mean, at some level, I would be interested to know what the answers are, or at least to have a sense of what we might want to think about with each of these. So, so the second question is containerization a good proxy for provenance? Are there probabilistic computational steps, e.g. random seeds?
Okay, I guess they're thinking about provenance for what, you know, when you run tools pipelines against data, keeping the, I would say containerization is not a proxy. That would be my answer there, but it would be a step towards trying to, you know, make things common and use the same tools, but you still have to keep track of the details of what the tool is and what the tool did when it ran. You know.
So, but presumably that all gets captured in a container, though.
Well, you can, but it's not. Oh, if it's just because it's containerized, and even if it's using the same kind of like Docker or something like that, doesn't mean it's going to collect the same kind of data, same kind of information, or the kind of information you need. So we still have to conform to both the, you know, the standard and collecting certain the data elements that we're looking for. You know, who ran it, what time it ran, what's the software called, what's the version number, those kinds of things.
Okay, but is that not all to some extent embedded somewhere in the container, or at least you know at least the versioning of the software and some aspects of that?
It doesn't have. Depends on who made the container, who created the container. You know, so you know, maybe if we had standards of how we set containers up, then yes. You know, okay. But you know, for HubMap, we're, you know, we have all the pipeline people off making tools and creating tools and putting them in containers, and we're telling them, okay, conform to this when you put it in the container.
Yeah.
But other groups may not be doing that same.
So are there a set of standards then around containers, you know, beyond what's required, for example, for someone to run on Docker? I mean, for example, are there kind of best practices in the containers world?
Around provenance, I don't know of any around provenance. I mean, that could be something that we could contribute to.
Okay.
You know.
Because you know, I mean, at least from my perspective, I think this is going to be an increasing challenge as people try to make their workflows available as containers. You know, will be this question of how to capture the metadata about the container, if that makes sense.
Yep. Yeah, that's exactly what it would be. That's exactly what we're doing there. Yeah.
But presumably some of that could be extractable in a machine learning or machine interpretable way.
Knowledge, yeah. So there may be. I honestly haven't done an exhaustive search for any standards there. I mean, something may exist I don't know about.
Okay.
but I'm sure there is some literature even there that I haven't seen, you know, even if aren't standards necessarily.
Great. So I think we've maybe a quorum of 4 at this point. We seem to be going slightly up. Would you guys like to quickly introduce yourselves?
Hi, Richard. This is Satra. I just closed down 8C. I was going to do that. It seems like there's a very small following on these subtopics. So
yeah. Okay.
I can update you on the other groups. This is the last one I was getting to. I'm sorry, is this A or B?
This is B.
B. I haven't gotten to A yet, but E and D had a fair amount of people, 10 or 15 or something like that.
E and D.
Maybe a third were active participants.
Okay, so maybe then we should split and join E or D.
It's up to you. One of the other groups that was small decided to make some progress first, and then they wrapped up to join one of the other groups.
Yeah, I mean, if for nothing else, I was interested in the answers to these questions purely for my own purposes.
No problem. Whatever you decide is fine. If you do decide to close out this one for B, just let me know and I'll make it unavailable so no one comes in.
Great. So for you two guys, do you want to talk through a few of these and see if we can at least jot down some notes on them? Do you want to go in the other conversations?
Would be happy to. I mean, some of these things are kind of relevant to us on a bunch of different things. Bill and I were just on the data movement and storage thing in the morning.
Right.
And a lot of these things, although this is under multiplexed molecular profiling, are kind of related.
So, I mean, what would it take to take a container for some kind of analytical workflow set up by one program and get it to run on someone else's data, presumably using a different infrastructure?
So there are solutions around it. I think a lot of it boils down to standards around both the data and the container. So it's kind of an impedance matching thing that if the container knows what to work on and that's standardized, then it will run on whoever has that data in that standardized form. And that standard could be a file system layout, it could be an API or some such. But yes, it's a current challenge because almost everything is kind of created in a cottage industry style. So I have my own layout. So this is where I think a big question for consortial approaches like this is to standardize both the data representation and what the container seeks to operate on.
So do you feel like the standardization of the data representation is further along than the layout of the containers?
I think so, mostly because most people don't think about the analytics side a whole lot. It's the experimentalists think about the data a lot more than the container. It's like a tool that they use to run on things. So as long as there's a tool that exists that runs on things, that's great. I think for consortial things, we just spoke this morning about making common pipelines that can be shared. And there's a little bit of effort on that front. I know CWL and other things have kind of tried to do a little bit of that, but it's not large scale enough across all possible analytic things.
Okay. So is it just by the very nature of containers that they can be designed to do basically just about anything? Is that part of the challenge here?
I think that's it. I mean, it's nice because you can package everything together and make it such that it's easy to take a container and run it anywhere you want. But from what Satra was saying, it's okay if the data isn't there in a standard format that it knows about to run, then it's not going to work, right? And so, but because they're so flexible and they allow all these, you know, the containerization technology is so flexible that it allows all these pieces that you would normally have to jump hoops to like do some installations and get a piece of software working for yourself. It's all in there. Basically, you can, you know, have almost anything installed in there, running in there.
Yeah.
So it's the, you know, it's the details of the software that's running in there basically is what that needs to be standardized along with the data, the input to it, which is data and metadata. You know, it could be producing metadata as well, and it's producing provenance information.
And to add to that, I would think that part of the role of a consortial approach is to provide standards and best practices. That's how you kind of get to people using the same things.
So do you think it's possible to scope down what would be done in a container so as to at least have a small universe of options for the format of the data going in and the format of the output of the black box? You know, I'm just using black box as a way of characterizing what the container is here, because, you know, to some extent, presumably it doesn't really matter what happens inside the container to some extent, as long as alongside the outputs we get some kind of output of the metadata of what happened inside the container. Is that a way to think about this?
I would put it at two. So I like the black box analogy as a starting point, that a person does not directly need to consider what's inside the container. It's kind of, I'm going to drive a car. I don't really know which engine is inside it or maybe I do need to care about what oil is being used. But that's kind of the analogy, that it performs a function. And so from a certain perspective, part of it is the description of that function that should be made clear. I work on a BAM file to do X, for example. And that's what's missing so the way analytics approaches work in a lot of labs is it's kind of handed down, almost like knowledge from father to son. You do this because that's how we've been doing it. And I think that's a place where a registry of containers, and the GA for GH is trying to do some of that amongst other places. That starts helping.
Yeah, the registry would be is a very helpful.
So is GA for GH setting up a registry then?
Yes.
Okay. Sorry, I'm just taking notes as we go on.
No, I think those are some of the things that are challenging right now, is that there's a lot of word of mouth kind of handing of here's how you do analysis. But for consortial things, and I think I would love to hear what HubMap is doing, for example. Do you guys have a registry of containers?
We have our own homegrown thing right now that isn't, I mean, it is open to the world, but we're just tracking these things in GitHub right now. So it's not really a formal registry. So if you Googled, container registry, you probably wouldn't find us, but we're effectively doing that right now.
Yeah, and a registry by itself is not always very helpful, right? So Docker Hub is a registry of containers, but you only download things that you know about. So I think there's the search part of this, which is I need to perform this operation. What containers help me do that? This is the kind of...
Right.
So we need to have some kind of functional description of what the inputs are, or something about the function of the container, and presumably some kind of description of the outputs.
Yeah.
As a way of trying to identify.
In a way of finding the container and more information on it, then I guess with the container itself, you would have the detailed information about what the exact inputs and outputs are, and what the what the churning on the inside is.
Okay. And is this something that GA for GH has discussed?
Based on some of the elements, some of the people who are involved with it, there is a lot of existing work on ontologies and efforts to kind of represent information. So EDAM is one of them that they've been using as part of CWL. And so I think there's a difference between the specification of something, which is what CWL is good at, and then the description of that specification, which is what this thing is doing computationally. And the latter is harder than the former.
Okay.
So that's something that we need. That's something we need we need in HubMap right now, is that description of what's going on inside there. And people have, documented in some cases very well what goes on the inside, but there's no standardized way of documenting that right now that I know of. I think we're trying to throw everything into, if you have documentation for this kind of thing, we're trying to get it in protocols.io. But there's still no, even within that, there's no standard way of, specifying, these are the details in an organized manner.
Yeah, I jokingly say sometimes now that the biggest challenge we face right now is the information filtering problem. It's not the lack of information, it's how to search through and find relevant pieces.
So should it, maybe as part of submitting something to registry, could be to fill out a certain minimum number of fields about, that provide descriptors for what the container actually does?
Yeah.
Okay.
Yeah. And one can easily see that in the future, you could have some machine learning technology or other thing kind of doing this, but that's not ready yet. Right now, I think the human is still the person who constructed that container and put things in. And I would say not just a container construction is relatively easy. It requires some system level knowledge. It's the analytics that are in there, the person with the kind of link to domain knowledge that said, I want these things in a container. That's the person who would be most, it's like writing a method section in a publication. Right? Yeah. And at the very least, if somebody could say, this is what it does, that becomes a starting point for search, which we don't have right now.
Okay.
But if you standardize what inputs goes in, what the description is in some ways, and what outputs goes out, that gets you a long way. And even with some smaller scope standards, it doesn't have to be the full detail to start with.
Okay. So is there anyone else working on this apart from GA for GH that you guys are aware of? You mentioned, Bill, that, it's something we've been struggling with inside HubMap, but are you aware of any other efforts that are going on, people struggling with the same question?
I'm not. I really haven't had the conversations with other folks.
So I can say,
I'm sure there's something out there. There are other people. I'm sure there are other people working on this stuff. I just haven't in the search.
So my group does a bit of this in the neuroimaging space, not necessarily in the cell atlasing space, but in the neuroimaging space. There's also an effort from Carol Goebel and her group out of University of Southampton. She was the original creator of Taverna, which was one of the early workflow systems. So her group has done a lot of work in this space, and they're associated with CWL as well, and GA4GH. So some of those things are places to integrate as a consortial effort to try and do this.
Okay.
But I think some of this comes from one of our target items that came out of the last thing that we did this morning, which is once you start getting to common pipelines, that's a good place to kind of intercept this and say, when we make those common pipelines available, can we put all these pieces in together?
Yeah. So from the conversation this morning, was there a natural starting point for this? For example, the single-cell RNA-seq data, or is there some discussion about, what may be a good test case for this?
There was some consideration of BICCN and HubMap, because BICCN has its brain initiative, but it has a lot of RNA-seq, transcriptomics pipelines, and which would kind of, I think, apply to HubMap as well.
I think we left that with a pretty definite plan to get together to work on these.
Great. And presumably then all of these questions around the descriptors for containers are going to come up pretty quickly.
Yeah, this is the next phase, stage.
Okay.
Yeah.
Okay. So it feels like we've beaten that one to death, and it sounds like there's at least a plan for how that might move forward if there is some space for collaboration between the ICCN and HubMap. The next one I was interested in as well. So how would you evaluate reproducibility?
That's a tough one, because that's part of the reason. So when we created this subtopic, we considered that matrix, right? That we can think about re-executability. That's an easy thing. Same data, same analytics, repeat.
Yes.
Robustness is same data, different similar analytics. Do you get the same outcomes?
Yeah.
Replicability is similar data, same analytics. So it kind of helps partition the space of reproducibility into a few components. And then generalizability is what we all want, which is as long as you have similar data and similar analytics, you should still be able to make the same claims.
Okay. So can you say that again so I can type and write it down as well instead of understanding it? And
I can actually post an image that captures that in there.
Perfect. Yeah, because this has come up in some of the experimental discussions about what are the different types of assays to understand different sources of variability. So a slightly different way of framing the question, but it's sort of, I think it amounts to much the same thing. What are some of the factors that, or some of the parameters that vary in an unpredictable way versus, what can you control for in different experiments? For example, what happens if you run SRAD version 2 versus SRAD version 3? So, I think there's things like that, whereas there are things like random number generators, I think, as you pointed out, that may influence the results at different time points, or parameters for clustering, or whatever it is that may be more on the user side or machine generated side.
I can't seem to paste or write anything.
Okay. Oh, I think, you can.
Yeah, I can write, but pasting, I think, may have been disabled. I don't know.
Possibly.
I'll see if I can do this. Okay, I can at least leave a link to it.
Okay, great.
So yes, again, coming from the domain I come from, everything matters. Operating system level things, libraries that a given analytics depends on. And so we are kind of looking at this as what are the different sources of variance to the output. And some of it comes from the data set, obviously, but a lot of it also comes from the analytic processes. Mm-hmm. So part of us breaking down this into these components was to target different pieces of sources of variance, right? If you keep the data set intact and you just move around the analytics, which are supposed to be similar, you can capture how much of the outcome. It's like measurement precision for an instrument, and you treat the analytics as an instrument.
Mm-hmm.
And the generalizability piece is where, as scientists, we want to get to, that the claims we are making are independent of data sets and analytics. Mm-hmm. And that requires replication and robustness.
So, I mean, are there, you know, what are the equivalents of a technical replicate study or a biological replicate study? Is there, you know, is there ways of thinking about that? And
some of the ways we are thinking about it is we are using the term vibration testing a little bit, is how changes in parameters affect the outcome. And so that's a way of looking at, if a finding is robust, you know, slight changes in parameters are not going to affect it. But if that now wildly changes the output, then A, you understand the sensitivity of what parameter of the analysis is related to the outcome. And sometimes it's clear, but in general, we're dealing with fairly complex workflows if you go from data to scientific claim at the end of the day. And so there are many parameters that come into play, and I don't know much about the molecular profiling world, which is why I'm a poor substitute there. But almost everything we look at in terms of analytics has parameters, and those parameters are often tuned to specific data sets or assays, and somebody will take it and apply it to something else, thinking it's similar, but we don't necessarily have the testing or the robustness behind it.
so, I mean, is there a set of generalized assays or approaches you could think about then for testing different aspects? I mean, presumably, you know, there's something about the noisy, like how noisy the data is, or something about the shape or distribution of the data going in. So, I mean, certainly in the, you know, the image analysis world, you can do things like blurring or add noise in and test the robustness of whether it's like feature recognition or not. So are there sort of generalized approaches like that that would be applicable across many different types of data?
I think so. Again, I think some of this would have to be data type specific to a certain extent. So in one of the other, I think it was 8A, one of the discussions they were going to have was on sparseness of information, right? So you can imagine different levels of sparseness, and yes, you have imputation and other strategies that one can bring in to bear. But probably at some level of sparseness, you simply say, I'm going to discard this sample.
But presumably there's probably an underlying basic information argument there about, you know, the amount of signal versus the state space or something, or you know, I don't know how exactly that would be described, but presumably it's more of an information problem there. Then
I would, it's you can kind of say number of bits, right? That and you can couch it that way. But I think real life workflows don't want to think about bits. They kind of want to think about which analytics kind of apply to what kind of data. And this is where people are doing quality assurances or other kinds of visualization to see if something is an outlier, right? Unsupervised dimension reduction approaches. So that will pick out some kind of outlier space alongside this. But there is, I think, a more systematic approach to testing of pipelines and then kind of testing it across data sets. Right?
So in your dream world, if you were to be able to systematically test a pipeline, what would you need?
Compute resources. I think, and data. So diversity of data is probably the biggest helper to an algorithm. It's not just the deep learning world, it's any algorithm. Because you get more robust, this is kind of saying that this works. And that also helps us consolidate pipelines, right? It tells us there does not need to be 10 different pipelines which were built to deal with specific quirks of each chip. We can now kind of abstract the quirks out to a single standpoint and then have a robust algorithm work on top of those. But there's always a tussle, and this again came up in our other discussion, which is the world of science is where you have stable things that we understand and then novel things that new technologies kind of bring to bear. And that becomes an exploration space where people create all kinds of hammers to understand. Can we try and bring those together?
So do you see a distinction then between systematic testing of an individual pipeline versus comparison of different pipelines for doing nominally the same function? Or, you know, is there a way to think about both of those together?
So in our world of those terminologies, those are slightly different. So one would be called robustness. The other would be looking at replication, right? So the single pipeline thing is where you try that single pipeline on different data sets that are supposedly similar and try to get the same outcomes. Whereas the other one, which is many different pipelines that do the same thing, you can obviously do both and both, so many data sets, many pipelines, but you can also take single data sets that you understand what it should be the output of, and apply those multiple pipelines to see how robust they are. In the ideal world, it will be a many-to-many map.
Why?
Because you want to understand if a particular pipeline is very sensitive to a given data set, right?
Okay, yeah, I see what you're saying. Okay, yeah.
Or an instrument. So I'm kind of using data set as a general term, but often it turns out it's specific to instruments that it was calibrated on.
Yeah. So Bill, has there been much discussion in HubMap, I should probably know this, about either of these aspects? You know,
certainly early on when we were discussing FAIR principles, so some of this stuff came up, but this hasn't been something that I've been as much involved in since then. So there very well could be that I don't know about. You know, on the analysis side, the CMU folks might know more about what's going on here.
Because I do remember at one point seeing a comparison of the different single-cell RNA-seq pipelines, and I think some folks in the Human Cell Atlas had done that comparison. Because, you know, they compared Cell Ranger 3 and Optimus, and there were a bunch of other pipelines they had compared, but I don't know all the details of that, and it seemed like, you know, that was a bit of a systematic study to understand some of the bias that each of the analysis pipelines introduced, in that they, you know, they seem to work with different types of data in a particular differently. So sorry, I think it was the expression of particular genes because of some of the parameters that are set in the way that they did their reconstruction of the data. They handled genes that are expressed at low levels differently. And that sort of seems to tie in. It's not quite robustness, but is there a different way to describe that? Is that sort of parameter testing, or is there a phrase that's used for that?
I don't.
Sensitivity?
Sensitive, that's like parameter space.
Yeah.
Because I mean, you know, I suspect that's one of the challenges that seems to keep coming up with things like cell segmentation and images. You know, a lot depends on what you expect to find in the data and what your, you know, eventually what your use case is for the data downstream, because that'll tweak exactly what starting parameters you might want to use. But it also seems to vary according to, you know, the algorithm as well. So it seems to be this sort of complicated set of options that each lab has found what they're comfortable with, but it's hard to make that portable across labs. In part because,
but it's partly kind of consolidating that knowledge, right? That's perhaps what a lab can produce. It's often that's the part that's hidden behind the scenes because they've now gotten to a scientific claim and don't think about all the processes that they undertook. And I think as Bill was saying, kind of converting some of this to a protocols.io type of approach, saying, we did this with these parameters. Now, why did this lab choose these parameters specifically? I think that's the level of knowledge that would be nice to consolidate at a consortial level.
It's nice actually using that tool, protocols.io. It's nice because you can define an analysis technique and then run it. If you change some parameters or change something, you can specify that and save it against that same thing, and it gives you that instance of what your analysis was, that very instance that you ran.
Okay.
So that's nice. That's actually a nice getting to the technical how we would do it kind of thing.
Yeah. Cool. And I feel I knew much more about this than I did when I started. So it certainly feels like there's something to pilot here, potentially. You know, I'm not quite sure what it would look like, but you guys probably have a better idea based on the earlier discussion about, you know, it seems like there's two things we need. One is lots of data that we understand the details of, you know, both in terms of, so let's say different quality measures of the data, but, you know, having that both diversity and the breadth of data seems like an important part. And then the computational resources seems to be the other part. But is there anything else that at a higher level we would need?
So you brought up something that I think is important, which I don't know if this field has a lot of handle on. It seems like this is different across fields, which is quality of data, right?
Yeah. So could you say a little bit more about that? And
so question is, what constitutes good data for a given algorithm, right? So it's one thing to say data is in a given standard. That's the easy part, I would say. And then you run it on that. So it almost seems like there has to be some set of computational processes that describe quality related to data. So amount of missing data. And it could be a vector. It does not have to be a single number. It would be nice if it was a single number, but I think it really will be in the eyes of the application. So some aspects of noise can be ignored by certain algorithms. Some aspects are detrimental to those algorithms. So even just knowing that kind of impedance match of which aspects are going to hurt an algorithm would be a good kind of trigger point so that people don't use things blindly, which is, I think, often what happens. As long as I'm getting the result I'm looking for, I don't question what I have done.
So there's, or at least there's people who have thought about these general quality measures of data, and you know, whether it's something more basic like a sort of signal to noise ratio, or you know, then there's more complicated ways of capturing this. But I mean, it doesn't seem quite like what you're talking about, though.
I think it is that. So maybe I can ask you guys, as you have processed data in your life or seen processing of data, how would you say the quality assurance or the quality control process has happened at the analytic space? I'm not talking about the biological data collection space, but once you've got digital data, do you consider all to be really good data? Do you do certain things to say, no, I'm expecting this data set to have had this expression, and it did not?
Yeah, I mean, in my past life working in the biostatistics department, I certainly saw a lot of, you know, looking at the data before you run an analysis on it, and then looking at the output to make sure that it makes sense with different measures and things like that. I've seen a lot of that on it. I myself am not a statistician, but.
Could you say a little bit more about that? Because, you know, again, my basic understanding was there's a bunch of things that are independent of it's at least somewhat independent of the data. So, you know, if you do sort of like, so if you look at the intensity ranges over an image, and you know, there's not much variation in the intensity, you can say something about that. Or, you know, if it's saturating, you can, so, you know, there's those types of measures which are somewhat independent of, I mean, I guess they're linked to the data type rather than the, what exactly you're looking at. And then there's the ones that depend on what was actually in the image. So, you know, you'd need to segment out the cell and try to classify the cell and ask the question of whether that cell contains the data you would expect. You know, either there's some missing data in there or things that have been misclassified. And so at least my understanding, there were those sort of two general sets of quality assurance that you can do. Is that, am I just making that up?
Seems reasonable to me. I'm not the right person to ask, certainly.
So are there protocols in HubMap, for example, that do quality assurance prior to any downstream analytics, or is all quality assurance based on downstream outputs?
No, I think there's definitely quality assurance that happens at the data producer, both at the, you know, the raw data, the time that the raw data is produced, and then along the way.
And is this somebody visually looking at things? Could this be computationally modeled? And if so, is that an essential kind of computational pipeline that could be applied to all data sets? For that data type, I should clarify that.
So at least my understanding of the conversation so far is nothing complicated has been discussed within HubMap. I mean, I know that we've occasionally gone there in discussions, but I think most of, and Bill, please correct me if I'm wrong because you're involved in a lot more of these conversations, that you know, most of our quality assurance has been at a more basic level when we've talked about it. So it's more about, you know, is everything there that we would expect to see, and you know, that type of quality control rather than quality assurance, in that we're, you know, there's no deep inspection of the data itself.
Yeah, I think there's just so many different kinds of data that are being generated now in HubMap. So many different, I don't know, we had for the first data release, Peter, what are like 37 different assay types or something like that, and then there's multiple things underneath those. But you know, when these discussions, I've pretty much been hearing what you just described, Richard, is the QC kinds of measures, make sure the stuff's there, should be there, things are in the range, those kinds of things. But I've also been hearing like of discussions that I'm not part of that are more with the folks who are generating the data, that kind of stuff is on their side, that they're doing this deeper dive. They have the experience in the biology that it's coming from, and but again, those efforts, I don't know what's going on. So what I've been slacking with Matt Ruffalo at CMU here, asking him to join this group. So after break, he'll join this group if you want. So I think he has a little more insight onto some of this than I do, but he's going to probably say, oh yeah, I'll go talk to so-and-so at Stanford, or go talk to so-and-so at, you know, at Florida, or go talk to, you know.
That would be the fantastic thing that would come out, which is kind of a consolidation of those knowledge nuggets or pieces that everybody could reuse, right? So as opposed to just the person at Stanford or just in HubMap, right? This could go across consortia, saying, here are the ways in which we can associate different kinds of quality assessment of different data types. And for multiplex tools, I think that becomes quite important, that you're getting all these different tools There is the biology piece of this that people are quite familiar with. Can we translate their insight into a quantitative measurement of quality?
Mm-hmm.
And keep evolving that. I'm not expecting it to stay static as we increase our knowledge, but that might become a good way of characterizing these things. I'll give a silly example. We often use SNR, right, for many, many different things. But that's just a number. And I think what I'm hearing is there's a lot of biological insight that is also attached to some measurement level noise or variance.
Yeah, and just to as you can tell, I'm much more of an experimentalist. So in the experimental world, there's a simple measure of looking at quality of tissue using a bioanalyzer. So you can measure the ribosomal RNA content of a piece of tissue, and it gives you a number which people then take as a general assessment of the integrity of the RNA in that tissue. But part of the challenge is it's ribosomal RNA as opposed to mRNA. It varies from region to region depending on which cells you're actually sampling. So, it seems like whatever parameters you pick, if they're somewhat easy to generate, and they're particularly at a global level, people are always going to come back with pushback and say, well, here are all the reasons why that particular quality measure doesn't apply. And quite often those are the biological circumstances. Like there's a lot of enzyme activity, or this tissue is rich in a particular cell population that has a much more stable RNA expression profiles. So I think that's part of what I'm curious about here is if we do come up with these kinds of quality measures, does, apart from that being an activity in its own right, what does it ultimately change? Does it make any, and this is to your next bullet point, does it improve the trust in the computation side of what the consortiums are doing? If we can come up with some kinds of performance measures or quality measures for pipelines, does that really improve trust or not?
So I think it goes back to the greater, which is a scientific claim I make. I think that's what as scientists we want to be able to say, right? That this claim holds under a broad set of circumstances. And that could involve populations at the sample level. It could involve analytics at the analytics level. And so to be able to say that we've done this holds, that makes the trust in it higher. In the context of quality measurements, you're absolutely right. I think different people are going to look at different measurements. But what you said, this is because of more enzyme activity. That may be obvious for someone at some part of this processing chain, but completely non-obvious to a person downstream from it. And so having a set of measurements, I think, just provides visibility to these issues. Okay. I think there's a different step in terms of quality control, which is how those measurements are going to be used to either discard samples or discard measurements, et cetera, from future analysis, right?
Yes, yeah, that outlier problem. Oh, I think we...
Oh, sorry, did you guys lose me for a bit?
Yeah.
Okay.
Yes.
I was just saying that one should decouple the measurements of quality from how you use those measurements to do downstream inference or processing, because that might be application dependent. Somebody may actually be doing something that looks at, I want to look at all the noisy measurements and do something with it.
So I won't even try to recap what we discussed, but we talked quite a lot about thinking about provenance around containerization and some of the discussion. We had quite a bit of discussion there, and in particular how to capture some of the details of both the function of a container as well as things like its data inputs that it would accept and outputs, as well as being able to capture some kind of description of what happens within the container. So that was a nice conversation we had. And then we had our second major theme was talking about how to evaluate reproducibility, particularly of different types of data, and what systematic testing of an analytical pipeline look like, and how to do comparisons across different pipelines. So I think that was more, and we talked a little bit about quality assurance and what kinds of quality measures are there that are out there, and part of the reason for trying to define what quality is, and does that really improve trust in the data, for example. So hopefully, Matt, that gives you a very quick snapshot of what we spent the first 45 minutes doing.
Yeah, that seems quite informative. Actually, especially reproducibility. I'm remembering a talk that James Taylor at Johns Hopkins gave at CMU a while ago. He's involved in the Galaxy project, very heavily involved. And he had a lot of input about how difficult it was to get some of these tools to behave reproducibly. Even when they are supposed to, they often do not. But I think that's not been as much of an issue for the things that we've been using specifically.
So could you say a little bit more about the RNA-seq pipelines, and any discussions you've had there related to reproducibility? Do you have any generalized assays you try against the pipeline to determine how reproducible the results are? What are some of the challenges you see?
So the pipeline itself is completely deterministic. So that being reproducible in itself is not much of an issue. It's really, since we're using salmon for RNA-seq quantification, which is alignment free, it does give slightly different results than, let's say, Cell Ranger would in the same inputs. But it's really just discussions within the sequencing data release team of, is this margin of difference acceptable? Is this outside a range that they would expect? Is it still like, is the number of cells being off by 4% between those two analyses, is that acceptable to the TMC? So yeah, we're actually using salmon for all of the RNA-seq assays we can. We modified salmon for additional assay support. I think there were a half dozen assays supported. Now it's plus the few that weren't already implemented.
So maybe to dig into that a little bit more, one of the things we talked about is for some of these quality measures, and I'll pick up on the 4%. Is it, how do you think about that 4% in that, is that against the overall general population of cells? In which case, maybe 4% isn't that significant. Although, it could be one particular class of cells or cell type or cell state that you would end up having variable information for. So, what level have you been thinking about this so far? Is it with the context of the data in mind, or is it just with thinking about this in terms of data type?
So in terms of, let's say, is a certain type of cell more or less represented in our analysis versus what's done at the TMC? We would love to do that type of comparison. We do not have cell type information from any of the TMCs. What we might have from one or two of them is ad hoc and not really applicable to the full data sets that they've uploaded. So it's really at the level that we can, which is number of cells, number of genes with, let's say, enough reads mapped to them across all cells. It's really at a data set specific level without anything about cell type. And let me pull up some of these conversations to remind myself.
And would it matter? And again, this sort of came up in our earlier conversation, thinking about something like a measure of signal to noise ratio. It's a general measure for whatever kinds of data, but it also depends on how you define the signal. And what is the information that you're trying to extract, and at some level you can make that generalizable in that signal is information or information content, and then on the other side you can argue that it's very case use dependent, or it's very dependent on some understanding of the biological context as well as what you want to use the data for downstream.
Yeah, like so we would love to do, especially the biological context of some of the quality control metrics. What we mostly have is comparing to count matrices, and it's mostly like a threshold of whether a certain cellular barcode is too noisy. So we are going as deep as we can into the specific data types, but like I said, we're missing a lot of what we would want to go even deeper.
Okay.
I'm sure that didn't answer your question as well. What did I just do? I clicked a button that put an icon next to my name. There. I'm sure that didn't go into as much detail as you were asking about, but.
So just to follow up on that, Matt, I mean, you mentioned 4%. Is that out of a hat? Is that just like P less than 0.05? Where does the number?
I did pull that out of a hat just as an example of the magnitude of difference that we might see between, let's say, cell ranger versus salmon quantification.
Okay.
I'm pulling up one of the loaded data set information spreadsheets from HubMap.
And then, so, is that 4% based on one run? Is that an average over runs of multiple data types? Is there a way that you've been thinking about how to assess this? Is this just based on data from one lab? Because, again, one of the things we were talking about earlier is what kinds of diversity of data would we want as input into some kind of systematic testing of a pipeline?
So I'm pulling up the loaded data set information now.
And how might we translate some of that to standards and best practices that could be applied across consortia?
Yeah.
Yeah, so especially for us, the most comprehensive comparisons we've done are 10x single-cell RNA-seq, and that's all from University of Florida for HubMap. And that's across thymus, spleen, lymph node. I think those are the only tissues.
Okay.
Actually, yeah, and the number of cells for each of these data sets are compared individually. The number of cells returned is on the order of, I think, as low as 7,700, as high as maybe 21,000. And we typically see differences of 100 or 200 cells from from barcodes being too noisy or things like that. And this actually does get into a lot of tissue and especially tissue-specific quality control thresholds, which are necessary. Because in talking with Megan at University of Florida, she has said repeatedly that, especially when you're sequencing, I believe, the thymus, you get lots of immature T cells. And if you do standard kind of normal filtering based on mitochondrial read counts, you will discard almost everything that is signal in that data set, because that is expected for those cells and not for much else in the human body.
Okay.
So that's farther from the generalization that we were just talking about, but it's a good example of the type of issue that we're hitting in this standardization.
But I think that's a very nice thing. So somebody downstream from these consortia who are using these cells and does a blanket discard is going to throw out signal, as you say. So if there was a place as a level of a pipeline which says, I'm going to look at this and says, oh, this has to be done at a cell type specific level, then that becomes informative to that downstream user.
And practically at the level of running the pipeline itself is making sure to expose that as an input of what kind of threshold do you want on this normal QC metric.
Right.
And to document it clearly that this is appropriate for this cell type because of this specific biology, if it doesn't apply elsewhere.
Right. And this can come from two different places, Richard. I know we are talking about provenance on one side and quality assurance. So on one level, you can kind of think of this threshold being recorded as part of whoever is doing the analysis. And that gives rise to a distribution of thresholds that people are using across different cell types. And that itself is a level of knowledge that kind of comes from the biology side without imposing something from the computational side right away. And that might lead to saying, okay, now this looks like, everybody, this is a very wide variance of thresholds being used for this data type. What is the reason for that? Versus there's a very narrow thing that's being used for this data type. We might be able to fix it. So I think there's two sides to it's a closed loop system that we can use to improve the standards and best practices.
So Matt, in your ideal world, what and do you feel like we've got a good enough handle on the performance of Salmon versus Cell Ranger 3 that we don't need to delve into this too much more, or are there particular kinds of other quality assurance checks you would want to do?
Well, so just in addition to normal things like running fast QC on the raw reads and reporting those results too, I think we have a good handle on the accuracy and especially the speed of Salmon compared to Cell Ranger. And I think we're working with some of the other TMCs for additional assays that we added support for, because that then isn't just comparing Salmon against whatever pipeline they developed. It's Salmon plus our modifications and making sure that those are not buggy, which of course they were in the first few iterations.
So have you compared?
Sorry.
Okay, so whenever you, so I'm going to try to parse out the different types of measures. So accuracy in terms of whether they're identifying particular, when you do the alignment and you identify the genes and their expression profiles, they are what you expect, or at least, they are. I mean, how do you define accuracy in this case? Is it comparison against the 10x Cell Ranger? Like, kind of how do you deal with defining what the ground truth is?
That's an excellent point. We are relying on the TMCs who are producing this data type to tell us what the ground truth is.
Okay.
And that assumes a lot about the accuracy and I guess there aren't many words besides accuracy. Accuracy of their pipelines, which, not to mince words, some of that is a grad student threw together some shell scripts.
Yeah.
So that's an excellent point to raise in terms of what we can compare to. And we pretty much have to work with what we have from the TMCs. We trust that since they're generating this data, they know what to expect.
And then we'd also talked about earlier, kind of the idea of robustness, and how robust is it to changing any parameters or altering the data slightly, or adding noise in, or do you do that kind of testing as well?
Let me see. I'm looking at there actually aren't that many parameters to Salmon.
And in terms of, as you said, getting these scripts from folks, do you have a dashboard that compares the outputs of those scripts across the different data sets that you get, as opposed to the specific data set that came with that script?
Nothing as formal as a dashboard, but we have outputs of running those pipelines on many of the data sets uploaded by our tissue mapping centers. And that's more of a manual comparison that we discuss with the staff at that tissue mapping center one by one.
So I wonder if in the theme of these meetings, if we've discovered a place here where cross consortia, we can help each other with this. If we're gonna, especially so you weren't in the meeting earlier, Matt, Satra and I were in, but we were talking about trying to use the same, at least a couple pipelines for similar data across consortia. And if we're looking at raw data as well and QA measures on that from across different labs, how is that done across different labs? It could all work into the same, sort of same workflow there.
And
it's
it
my
might
novice view of it, by the way.
And it might also help fit some of the things that we were discussing earlier in terms of standardization, right? So if we can get a registry of these Docker, so they may have shared scripts, we could Dockerize it, create a registry of containers. Once that exists, each of these consortia can run the same containers across their different data sets, pull the results out in a standardized format, perhaps create a dashboard of the provenance so that we know how to link back from those outputs. And I think that that's not technically too hard. It will require a few resources to get some of the things going, but I think that could help across consortia quite well.
Yeah, in fact, we have Dockerized and turned a few of the pipelines from the TMCs into workflows written in something like CWL. We did that just practically so we could run them on cluster resources without a lot of custom configuration on the cluster itself. So many of those are not as production ready as the pipeline that we intend the infrastructure group at HubMap to run. But those could be released with a little extra work to clean those up.
Yeah, that sounds like a doable action kind of across the consortia across a set of people in different consortia who might be willing to make a consistent registry. And the other piece, Matt, that we talked about that would be useful is kind of giving a description to the Docker container or to the container saying what it does. That's one of the harder things in terms of a search space for containers, right? If you don't know what you're looking for in Docker Hub, you will never find it. You might accidentally run into something. So kind of creating also that specification to help the person who's searching for. And a starting point could simply be text. We can do all kinds of structured things downstream, but even at a basic level, having a text description of a container would provide some starting point for search.
Yeah, actually, so the CMU tools component for HubMap is split between PIs at UCSC, the Sanger Institute, and CMU, of course. And we have a research programmer who's half-time on HubMap at UCSC. He's a lot better than the rest of us at adding structured documentation to the workflows themselves in CWL. So that's such a starting point of how do you, what's the input, what's the output, what language is it in, if that matters.
Right.
So maybe we can loop them into some of this conversation to talk a little bit about, you know, what are the important aspects to capture in terms of descriptors for the container, and to find a way of maybe providing those details in Docker Hub.
Yeah, that sounds very reasonable.
But
also, be... Doc Store, I think, is the combination of test data, the container containing the tool, and the workflow. And I think it supports a few different workflow languages. And the intention is that most or all of our pipelines will be published on Doc Store, which is a little more. People always lie when they say it's something is self-documenting, but the intention is for those workflows to be more easily usable just by looking at the workflow itself.
And I think that the key thing there is it's not, we've been talking about containers. It's more than one container, because we can have a CW workflow that runs multiple containers that are chained. And that's what Matt's been working on. In fact, Matt actually found a way to run CWL with CWL Runner that gives us provenance information out. So.
Yes, I found it. It was other people put a lot of effort into implementing it. I just found the command line option.
That's what we like.
Yeah, actually, I can share. What else do I have open here?
Yeah, Matt, on some of those fronts, would be happy to chat later on. I know part of this kind of follows up on things that we might be able to do. So we have a workflow engine that should support CWL as well.
Yeah, this is a single cell attack seek pipeline that we're developing. We're actually wrapping snap tools and snap attack. This is an example of the machine readable metadata that our developer at UCSC has been adding. So license keywords, author. Not too much, I think, in, oh, this is only one sub-step. This isn't the entire pipeline. Yeah, I'm not sure why those are commented out, but in principle, these are usable for searching in the way that we've described. Oh, and also it's worth mentioning at some point, I'm not sure this is the right time, but it's a time, that we, in addition to encapsulating tools in Docker containers, we build a container for the tool with whatever software requirements and dependencies, then extend that and add any supplemental data that is required. So that's actually baked into the container in terms of like the genome index, transcriptome index for salmon. We did this just to make things practically easier on the infrastructure group for HubMap. So the only input to the workflow is a directory. And that helps reproducibility a bit, except for the inputs to those containers that you then have to find if you want to reproduce the container itself. But the pipeline is, in terms of specifying not only what to run, but the data as part of the container, we find that to be quite useful. I think the main practical reason we did that was that dumping the provenance info from a CWL workflow includes all of the input files and all of the output files in that provenance bundle. So a duplicate copy of the build 38 human genome in every single output was not desirable.
So maybe I misunderstood what you said. So there's an internal reference or calibration against the human genome version 38. Is that so? That's included internally within the container. Maybe I misunderstood, Matt, what you said.
Yes, it is.
Okay. So the way I think about it is it's sort of an internal reference / calibration, but it's not really a calibration. It's more a reference.
1 second. Let me share this again.
I think it's a reference.
Yeah, so just as an example, oh, that's hidden. So this needs to move off of my work desktop. So we have a container, let's say, for the salmon tool itself, with our modifications for extra assays. Then the index, let's say for GRC-H38 for salmon, is included in a container that you can run. So there's no issue of which reference genome do I use to get the same results that HubMap did. You can use our tool. You can use our tool and our supplemental data as one if desired.
Okay. So that seems like a good best practice in terms of designing containers then?
I agree. And everyone else who I've mentioned this to says, that's weird. I've never thought of that.
So I think part of that might depend on whether the data is accessible or downloadable from a particular source and how large that data is. So that's another thing that kind of came up. So I think the key is that you have a versioned copy of the data or downloadable piece of that from somewhere, right? That the checksum can match up whenever somebody does that. So whether you build it into the container when you can, which for that file might be fine, but for other things that may be too large to include into the container, but as long as you can access a specific version that you can verify, would still result in reproducible practices.
So at least if I was thinking about it in terms of an actual instrument, there's some kind of power on self-test. So, I mean, is there something equivalent for a container from that perspective? And then, secondly, there's this question of, calibration. Are the results that you're measuring calibrated against some kind of external standard? And then, version control. There's a bunch of things that you would want to specify as part of determining the quality of information coming off an instrument. So, is there some way of thinking about some of those, some of that kind of quality assurance from an instrument in the same way for containers?
Absolutely. I mean, conceptually, I don't think there is any difference really. I mean, these days for a lot of software, you have testing frameworks alongside it. So if you think of the container as an instrument, it can have modes, if instrumented properly, to do all of the things you said, Richard. Right? It could run a self-test. It can give you quality measurements on external data if you want. But that's kind of the engineering of making sure. So just as Matt has done for his things, he's included this first step of having some of the data in the container. Right?
Oh, and also practically Docker containers are composed of multiple layers with each command that you run in a Docker file. Each of those is named by its checksum of the files that are the result. So I have never attempted to do a Docker pull from something that is corrupt to see whether the checksum is actually verified as it's extracted. But that's a possible layer of ensuring that there's no bit rot in the reference genome, for example.
Right. So the one thing, Matt, that would be is whether that URL that you're curling is fixed, right? Because that's a network operation with a namespace. So the endpoint of it,
the IEC, that's currently just on my work desktop. That needs a more permanent home by far.
Right. So that's the part which, if it was a checksum location where the link could be verified, then it becomes consistent. Otherwise, Docker will not pull that file the second time you run it, even if the underlying file has changed.
Actually, that's a great point then. So,
but these are all doable things. That's just engineering. But I think the conceptual framework, what you said, Richard, I think that's an actionable thing, which is to make containers that are distributed by the consortia adhere to certain common principles.
Yeah. Yeah, and I mean, there's always this balance between, you know, getting that consistency versus adding a lot of work on everybody's side. But presumably, if there are somewhat standardized ways of doing some of this, it's to go from one to a new variant or adaptation, you know, some of that can get copied across. So it may maybe work the first time around, but hopefully it is reasonably scalable.
And it is a mixture of those pieces, including the things that Matt was talking about in the CWL descriptors and other pieces. So it's not a single solution, but I think if we can get the conceptual pieces across to people producing these containers, I don't think from an engineering perspective, it's a lot of resources that's required to do some of these additional bits relative to the longer term usage of some of these containers across consortial projects.
Okay. So does Matt, I don't know if you know this, does DocStore have any kind of baseline requirements for when you're uploading something to DocStore? Is there baseline requirements there?
Yes, in short. So things are separated out as tools and workflows. A tool should be one repository that produces one container. That's actually not how we had structured things at first, so that was a bit of a pain. You must provide example input files, or at least links to those in maybe some cloud storage, so people could conceivably run the pipeline right away without having to search for suitable data. I'm blanking on what the other requirements are, but there are at least a few that are intended for people to get up and running with the pipeline as soon as possible, if they're interested in using it.
Okay. Because it, you know, it sort of feels like we should get those people involved at some point in some of these conversations to see, you know, is this something they've explored previously? You know, I mean, my impression is that they're primarily there to operate as part of the Cancer Research Data Commons. So I'm guessing there's been some discussion in this direction already, but it would be good to know, you know, if they have at this conversation and if they have run into any pushback on it or challenges or.
Yeah, certainly. I know Ben Patton at UC Santa Cruz is heavily involved in DocStore, and he's also one of the PIs for the CMU tools component. So
yeah,
that's why there were a lot of DocStore references, I think, in the response to the article. But yes, further collaboration with them seems extremely appropriate.
Okay. Was there anything else people wanted to comment on related to that? Because one of the things I'd love to jump back to is to talk a little bit about, you know, thinking about if there are particular kinds of data sets that we might want to collect potentially to understand some of these questions around robustness, or to be able to compare different pipelines, whether it's data sets generated in a consistent way but across different tissue types, or different data sets on different versions or different machines on the same tissue. Are there particular kinds of things that we might want to think about in terms of data collection that would be of interest in understanding the performance of some of these analytical pipelines?
So at what level are the current data sets public or not public? Let me start with that quick question. Because if the data sets exist, these tools could, from a dashboard perspective, be run on multiple data sets that just kind of measures the variance automatically. So are the data sets, Matt, that you're processing just stored locally? Because you said you're generating but don't have a dashboard. You're running these tools, right?
Yeah, the data sets that we're working on from the TMCs are stored on, actually Bill can elaborate more on how the infrastructure component of HubMap is storing those. The processed versions of those, we just have as one-off direct communication from our tissue mapping centers. So they're kind of on whatever machines we have to do that manual comparison.
That data is not public yet.
Yeah, just wait until June.
Okay, but it will be.
Well, so there's some question around the if it's raw sequence data.
Okay.
Maybe not.
Okay. So then it might be at the level of the consortium itself to run these kind of comparisons, if
it's not public data.
If it's not public.
But it might be across, one consortium might have to run it and another consortium runs it, and then they compare the results.
Right. And since everybody uses UMAP these days, they've moved on from TSNE. I'm just joking. But part of it might be that as part of the measurements that are collected from this, that could be used to create kind of orthogonal data sets that behave differently across these tools. And that might kind of bootstrap a set of different data sets that look different from a tool output perspective to say, why are these different? So that might be one way of bootstrapping those data sets, Richard, that within a consortia, it's kind of things that show variance for a given set of pipelines across data sets that are then looked at as for future tests for those pipelines. And then one can then kind of do a comparison of the data sets themselves as to what's different across the data sets across different consortia and tools.
So let me throw this out there because this came up in a discussion we had a month or two ago related to the different common fund programs and the data sets they generate. So the suggestion came up that we try to find a better way to handle data access committees, and in particular data access committees for different but related programs. So, for example, you can pick the BICCN and HubMap as examples. In theory, there's two different data access committees that control access to the raw read data. And that sort of makes it more difficult and complicated to get access to both data sets if you want to run any kind of joint analysis. So is there enough reason that we would want to try to change that system? Or, basically as you were suggesting, should we just approach this as each consortium runs its analysis of its own data in parallel, and then we compare the results?
So I think that's one. I would suggest that if we can, at the level of such a consortial approach, also consider where the consortium could decide on Docker containers to be sent to different consortia to run, right? So a two-way street that allows sort of a common set of Docker containers that run at different places in case access is not provided across. I think technologically access can be solved. It depends, given the international nature of this consortium, it probably depends on different data relocation governance policies in different parts of the world. We came across one in our previous conversation where data from India is not allowed to leave India. So those constraints are always going to exist. So I think the other way to think about it is bring the compute to the data, but compute not just what the consortium is doing, but maybe they have the bandwidth to allow for some additional vetted containers to come in and run things.
Yeah, and it's a question that comes up for us on a somewhat cyclic basis, because there are other alternatives out there, such as the Anvil platform that NIH-HCA has put together as kind of a workspace where people can work with data sets from with different provenance. So it was just a question to see if there's any particular enthusiasm from you guys for us to accelerate what that process might look like for changing data access or not.
Yeah, I come from the data archive where all data is open and public. So I'm a very different side of a data archive. So I'll leave that to other kinds of types which are not public to kind of make that statement. But the way we are approaching this in the BCDC, which is the Data Coordinating Center of BICCN, is kind of separating out components that require kind of authorization for access and components that could be made publicly available. But all of BICCN is essentially in either the US or EU, which has very different sort of rules and regulations versus other parts of the world. So we can do certain things at the consortium level for access, which may not be possible at the overall international level of consortia.
Okay. So
yeah,
one of the...
I just wanted to bring up one other thing. So for the BICCN, some of the things that's happening is NDA, which is the NIMH Data Archive, is going to house some of the more secure data, and through an NDA, we would get people would get authorization to access things at the given archive. So NEMO is a specific archive which is housing genomic, transcriptomic, and genomic data. For restricted data sets, they would get authorization through NDA and open up for access.
Okay, so your rights to access data sets because you were part of a consortium would carry over to NDA? Is that?
So that would be a conversation with Greg Farber and the NDA folks a little bit. I wish that were true. So, okay. But I think that's a different policy level conversation than I can speak to.
It might be a good segue into one of the bullet points we didn't discuss this morning, which was earlier on, which is where and how can multiplex data be made available. And this has come up at least a couple of times in the HabMap, given that we do have some unusual data types present, and at least in my mind, it's never really been clear how we make those available in the same way that we do for the more common data types. And I think that this is only likely to increase, that we'll have more of these multiplexed approaches. So whether it's integration of ATAC-seq and RNA-seq data, or we were talking earlier today about in situ sequencing and codecs. It sort of seems like there's going to be a proliferation of different types of multiplexed approaches. And is there anything particularly unique about those that we should be thinking about at this stage in terms of making them available? Matt, I'm not...
I don't know. So I think it would be great. It's always good to me to have data available. It helps algorithm builders test their things. So the question is, which consortia are generating it now, and would they be willing to make components or subsets of data available, at least at a starting point, for kind of tool development purposes or visualization purposes? That could be a common ground to help kind of say, oh, these different consortia are generating multiplex data. Having common tools that process/visualize the data could be a shared common space for these consortia to collaborate on. And that would also help iterate on the access process to such data. Now, I don't know how sensitive the remaining pieces of the multiplex data are versus the genomic components. So
I'm not
again,
totally sure I'm on the same page of what multiplex data means. Is that multiple types of measurements in the same cell?
Yeah.
Okay.
Yeah.
So I'm not exactly sure what in HubMap we have of that besides the snare-seq assay, which is a combination of ATAC-seq and RNA sequencing for the same cells. I think some of the tissue, well, I know some of the tissue mapping centers are taking successive slices of tissue that are maybe 10 microns each and sending different slices to different assays. But those aren't necessarily the same cell. Those would be, you might get the same cell in both slices, half of it in each, but that isn't guaranteed.
We may get some post-processing stuff on that data that we get data recognized as a cell across multiple assays, I think.
Okay, that's fair.
So normally the Caltech UW Tissue Mapping Center is supposed to generate combined immunofluorescence and RNA fish data. And it's supposed to be simultaneous. I mean, I think they've had a few issues in getting the right level of signal in both channels. And likewise, you know, they're not generating data at this point, but the group at Harvard, the TTD project at Harvard, are also basically doing the same thing.
I was just going to say that I have a lot of tunnel vision for what's going to be in the first data release. So if it isn't listed here, then I'm thinking of it for very near future, but not necessarily now.
Yeah, but you know, even for the snare-seq data, at least my understanding was that we were processing that as RNA-seq data, or at least the RNA-seq part of the snare-seq data. So we're at this point we weren't handling it as an integrated multiplex data set.
Correct. That's handling it as doing integrative analysis of both. That's kind of the very next thing to do after first release, or at least extremely close. Yeah, we're treating each piece separately for now. And
I mean, are you guys then thinking about different outputs from that analysis? The same outputs? Like, I mean, are you going to handle what happens with the data differently, or is it going to be pretty much the same, except trying to take advantage of the ATAC-seq data to help refine some of the RNA-seq, for example, clustering? Like, I mean, like, how are you approaching this question of, you know, is the combined ATAC-seq and RNA-seq data together, is that classified as a unique data set, or are you just handling it in the same way you would handle both of the different types of data?
We haven't had many concrete discussions about that, but using it just to, well, essentially giving more dimensions to a dimensionality reduction and clustering method is one possibility. Anything that's more biologically driven in terms of what we can do with that, I think we have some conversations scheduled with UCSD about what we can do, but I think those are on the back burner for now.
Okay.
So in short, not much concrete that we have discussed.
And I think Richard 8A is supposed to be discussing that issue of kind of combining these multifactorial data types, and even clustering on them becomes an open research problem in some ways.
Yeah.
I mean, you can do something bad and naive that would give results, but is it useful?
Yeah.
Right.
So just coming in to give a 20-minute warning. We're due back in plenary at 4:05, so we have 20 minutes or so left. And this group, as well as Group E, exploring and visualizing multiplex molecular profiling data sets, haven't really engaged. There's nothing in the blue box yet, so just checking in on these two groups to make sure you guys are good to go.
All right. Thanks, Costa. We'll work on that.
Did anyone come here from Group E? The room is empty at this point, so no? Okay. All right, we'll track them down.
Okay.
Take care.
Okay, should we try to fill in our blue box then?
Sounds good.
Okay, so we have to give a title. So, I mean, it seemed like we talked about a couple of different things. So, you know, we talked about this idea of coming up with better descriptors for containers and trying to make those available to make it easier to identify containers to run for particular types of data that people may generate. So, I mean, that seemed to be a whole bunch of maybe coordination activities might be the way I would classify those. You know, how to be able to collect those terms either from human input or from extracting information from the container itself, as well as getting that information posted on DocStore or wherever it might be. So it sort of feels like that's a bit of a coordination effort. Let's try to summarize that point.
I would describe it as containers and workflow.
Yeah. And then we had a second discussion around quality assurance and how to approach quality assurance of containers and workflows, and how best to I don't know the best way of describing this, test for quality, test for robustness, test for reproducibility, test for, there's a whole bunch of things that we talked about, and how to capture that information alongside the container in some way so that people can know which is the best container for a particular type of data input or for some particular desired use case or output at the other end. Does that summarize the other part?
Yeah, I'm trying to note down things that you're saying, Richard, so that's why my brain is slightly switched off. But yes, sounds about right.
And then were there other major themes that we talked about that we might want to capture here? And sorry, that last one, it sort of feels like the quality assurance part, you know, again, it sort of feels a bit like a coordination question, but there's sort of a couple more maybe collaborative aspects where it might require a couple of groups getting together to work on some of the details before trying to broaden it out to a wider group.
So standardization seems like one of the things that could be widened out, right? And I'm using the word standardization without any qualifiers because it seems like everything we talked about has a potential standardization component, whether the data interface, the containers, the container descriptors, and outputs and provenance, right? So,
yeah.
And standardization efforts are always tricky, but perhaps we can get low-hanging fruits to get people moving towards common pieces.
Yeah, and I think, you know, there's something about best practices, you know, back to what Matt was talking about at the start of this second half. You know, I think trying to capture some best practices around the construction of containers. I'm not quite sure where that fits in, but it sort of feels like it's not necessarily standards, but yeah, best practices.
And especially separating that out into practices for creating any type of Docker container, which is delete like the package lists after you update it through the operating system package manager. Okay, fine. That's mundane. Things about how to specifically make them better in terms of scientific workflows, like how much supplemental data should you include? Should you include any? Is that appropriate for what you want to do and what you want to publish?
Yeah.
I mean, on one hand, you'll be downloading 20 gigs from Docker Hub. On the other hand, you don't have to search for any supplemental data that's usable for this pipeline. So is that a trade-off you want to make?
So how does, this is going to be another very naive question, but how does version control work on containers? So, you know.
So the Docker file itself, it specifies how to build it. You version control that. And you tag the container with whatever tag seems appropriate. You can do like the commit from the repository that produced it if you want. You can tag it by date time. It's not tied together as well as it could be.
Okay.
And I think some of the troubling pieces are the ideal thing you would want is almost a bitwise control on the Docker. And that's actually much harder than it looks. So if you run the Docker file today and you produce a set of layers, it may be different unless you construct the Docker file in a way that maintains versions at every layer. And that's a much trickier prospect for the average container builder. Not impossible, but gets much harder. And that's the kind of thing that from a consortia perspective, maybe the published, polished containers would have that quality, but not every container to start with.
Yeah, so hopefully the font size won't be too small here. But one of the workflows using CytoKit for processing, or for doing segmentation and whatever for codex data, our release process as it is tagging a version for the IEC to use, which specifies a date and timestamped tag on that container.
Okay.
So this is specific. This will never change. That tag is usable in the repository. But yes, it's a bit ad hoc in terms of associating a tag or a commit in a repository with a specific container. It's whatever you write it to be, essentially.
Right. If we assume that containers are never lost, then you can fix a specific container with a specific tag. But if somebody's rebuilding from the Docker file itself has to be created in a way that makes the replication process.
So we do version images too, which is a built Docker.
Right. So as long as that's not lost, that's what I meant by there's a place to find that binary set of layers.
Right.
And maybe that's the other kind of thing that's associated with this, which is at a consortial level, a registry or an archive of those layers that exist. And if that's Dockstore, great. Put more resources into that to make sure that those are archived and people can always download it.
Yeah, we certainly do that in the Docker Hub. I suspect that Matt would know this. You can do it in Dockstore as well.
Yeah, Docker Hub, I have no guarantees of what the future will be.
Yeah.
Right.
Yeah, oddly enough, just in the last few days, I started to wonder where did they get their funding? How many bytes on disk does Docker Hub consume?
So is that essentially that fingerprint then what goes up on, let's just pick the HubMap data portal as example. So alongside any analyzed data set, that fingerprint for the workflow that is either related to, so it's related to that versioned CWL file or whatever it is that like how much lies behind what we would put on our website that describes the container that we use to generate the process data?
Yeah, that's a good point. So when we start thinking about, right now we're thinking more about data than the tools, but all the tools need to be part of this as well. And from what just right now, I just realized we got to keep those images those Docker images and the tool images, we can't just point them off to some website that we have no control over, right? So is that what you're getting at?
Yeah.
But we're not doing that right now. But we're not really, we're more focused on the data, the actual data right now than releasing the tools.
Yeah. But eventually, you know, I mean, I presume, at least for HubMap, that's the direction we're going, and most other groups as well, you know, as well as the raw data itself or the minimally processed data, to have some of that interpreted data out there along with the pipeline that was used to produce it, and capture the details so that if somebody does want to try to replicate it either with our data, but say with their own data, or to make a tweak on the particular analysis pipeline, you know, that there's some way of at least capturing in an easy format what we did.
Yeah, so I think that's one of the really strong advantages of Docker, is that if you have the layers, you can run the things. Again, it depends on the internals of the container. So if in the container I'm downloading data, now you have to rely on an external thing to ensure that you're downloading the same version of the data. So that goes back to kind of making the construction of the Docker image be sensitive to version control. But assuming that it's self-contained, which is, for example, the one that Matt showed, if you run it again, you still have all the pieces you need to run it. And so now you only need to make available those layers for somebody to reproduce. In terms of best practices, I would also suggest kind of embedding the construction of the Docker file into the container itself, which often many containers that are out there are missing. So that way people know how the layers were constructed.
So is there something like a RRID, so the, you know, kind of the idea of a research resource identifier for a container? You know, it gets back to this, you know, registry of containers, but, you know, is it possible to sort of mint some kind of for a container that's been used in, let's say, a published analysis of data that can then be referenced back to?
So I don't know if the doc store provides DOIs. So that would be the kind of thing that you would create.
Yeah.
So RRIDs have a slight disadvantage in this case. So we use RRIDs for software, for example, but RRIDs are not versioned. So they just kind of say it's salmon, for example, not which version of salmon.
Okay.
So a DOI could become a lot more specific, saying this is this checksum version of the container that somebody used. So now you can go back to that exact version of the container.
So would that be something worth discussing then?
So we should check if, I don't know if Dockster, maybe you guys know, Matt or Bill, that Dockster provides DOIs for versions of containers.
I don't know if they do. If they don't, from the HubMap side, we can mint a DOI for it. We're talking about, we're doing DOIs for data to data sets.
Yeah.
They do a DOI to a data tools, all the inputs in the tool as well. And that'll automatically be part of our whole provenance graph. If you click into any piece of data, you can see where it came from. If it came from more data in a tool, you can see that. So we can just put a DOI at that level to level up as well.
Yep, that sounds good. I mean, to me, a container is no different from data from a digital perspective. It's just another blob. No. And so you can refer to it through whatever ID mechanism we choose to adopt.
Yeah, I'm not familiar with DOIs on DocStore. In looking around at a few sample workflows and tools, I don't see much.
Because presumably, you know, a bit like the reference genome assemblies, you know, I think we'll have, you know, let's say versions of maps. So it would be good to capture how those maps were generated with whatever particular workflow we had at that time. And that, you know, it might be the kind of thing that would be good to have. I mean, certainly I don't think we should do it only for ourselves. You know, whatever we do as part of HubMap should be follow whatever is commonly accepted practice out there.
So I mean, as a follow-up thing, I can definitely, for those interested, I can definitely create a separate kind of side session workshop to kind of discuss best practices for versioning different aspects of an ecosystem.
Yeah, that'd be great.